Off the top of your head, what do you think some of the leading causes of death in the United States are? No, gun violence does not rank at the top, but cancer does, contributing to just under 600,000 American deaths. Now, cancer is the second leading cause of death worldwide, where in 2020 alone, it is estimated that there were 19.3 million new cases and approximately 10 million cancer deaths. Furthermore, this number is projected to increase by around 50% in the next 20 years. Cancer treatments have been steadily advancing, but current standard treatments are still confronted with multiple efficacy setbacks. So, how is the search progressing for effective, safe, and durable therapies? Hi folks, my name is Cole, and I have a Master's of Immunology. Today on Investigate, Explore, Discover, we're going to be looking at improving methods for adoptive cell therapy screening. So hang around with me throughout this whole video to get all of the relevant background information so we can dive into some exciting experimental results. Plus, there's more information for you in the description below. But in regards to cancer, we've known about it for millennia, though we've only really developed effective treatments for it in the 20th century. Even so, existing first-line therapies often fail to cope with the heterogeneity and complexity of cancers. In the 1980s, the search for safer and more durable therapies led to the discovery of immunotherapies. These approaches modify aspects of immune interactions with tumors. They can include using antibodies, cellular checkpoint inhibitors, or even something called adoptive cell therapy. Now, adoptive cell therapy is characterized by the isolation of cytotoxic immune cells from the patient. The cells are then multiplied in vitro, where they often have their function enhanced so that when they are put back into the patient, they recognize and can specifically kill the cancer cells. Lymphocytes, which are part of our adaptive immune system, are the cells that have been used in this capacity. Normally, cytotoxic T cells are used for adoptive cell therapy, but natural killer cells can also be used, which is what we're going to be focusing on today. Now, there are many different types of NK cells in our body, which we can identify through extracellular receptors like CD56 and CD16. All NK cells are cytotoxic, but some are more cytotoxic than others and are better at killing tumor cells. Under physiological conditions, NK cells leave host cells alone because the host cell has both an activating and an inhibitory ligand. However, cancerous cells lack this turn-off signal, so the NK cells attack and clear them. Now, one of the most well-studied activating receptors on NK cells is NKG2D, which recognizes the ligand MICA/B. This ligand is known to be highly present on cancerous cells and largely absent on healthy cells. Interestingly, tumors can avoid this recognition by cutting off this receptor with proteases, effectively evading NK-mediated clearance. Now, when NK cells get successfully activated, they release a whole host of molecules, many of which have anti-tumor function, like granzyme B, alongside pro-inflammatory cytokines like GM-CSF, TNF-alpha, and interferon gamma. Adoptive cell therapy is a promising approach for being able to deliver personalized care for patients, though this method needs to be rigorously screened because doctors and scientists are still ironing out the kinks. Some of those kinks are that they can be prone to cellular failure, cancer relapse, or host toxicities. Furthermore, this therapy is only consistently effective against blood-borne cancers, not solid tumors. Now, in attempts to identify and fix these issues, many models have been and are being used. The traditional models are 2D cellular cultures and animal models. Animal models are effective but costly and not always translatable to humans. 2D cell cultures also fail to mimic the architecture and cellular heterogeneity of a solid tumor. They also force cell-to-cell interactions, making it unsuitable to realistically recapitulate tumor immune interactions. In the past 2 decades, human cell-derived 3D tissue models have become more popular, as they overcome some of the simplicity problems of 2D cultures. This then gave way to microphysiological systems, which combine microfluidics with advanced tissue models. These systems can then be designed to mimic mechanical and physical cues of a tissue's microenvironment, making them key innovators to further our understanding of how cells function. Nowadays, we can connect up to 10 organ systems together for as long as 4 weeks, making them suitable for systemic investigations of intertissue communication and therapeutic testing. These whole body systems are so cool, but they're also very complicated and difficult to scale for mass testing. These complicated systems are needed, though, because the tumor microenvironment itself is a complex place. Tumor persistence is maintained through the inhibition of immune cell function by inhibitory receptors, release of cytokines, and by physically blocking immune cell infiltration with a dense interstitial extracellular matrix. The majority of reported immunocompetent microphysiological systems use cancers embedded in hydrogel, which mimics the protective extracellular mesh that envelops tumors. Though this model mimics the environment of the tumor, it really only allows for the readout of microscopy measurements. Furthermore, most of these methods only focus on treatment efficacy, which neglects to account for the possible off-target attack of healthy cells and the release of cytokines from the cells involved. This leads to the systems not being able to accurately predict what would really happen to the people given these treatments, which is something that I think everyone would want to screen out beforehand. Now, I want to take a moment and really highlight why improved immunotherapy screening research is so important. As it stands, there is a large gap between in vitro performance and in vivo efficacy when testing treatments. Advancements in multi-cell 3D models and microfluidic systems enable more accurate mimicry of physiological interactions of cells, which allows these models to better predict how well a treatment will function, giving rise to better therapies. Now, if you also think that these are some important reasons to research this topic, go ahead and tap the like button. This brings us to the paper that we're focusing on today. This paper is called An Immunocompetent Microphysiological System to Simultaneously Investigate Effects of Anti-Tumor Natural Killer Cells on Tumor and Cardiac Microtissues by Nguyen et al. From ETH Zurich in Basel, Switzerland. In this paper, the authors adapted a microphysiological system to allow for the immunocompetent co-culturing of NK, tumor, and heart cells, thus enabling multi-parameter study of the effects of the cellular interactions between them. To start, highly purified NK cells were isolated from umbilical cord blood. These cells are known to have potent anti-tumor activity in both direct cell-to-cell and indirect cytokine-mediated fashion. Now, the 3D tumor model was established from HCT116 colorectal tumor cells, which produce their own extracellular matrix and form solid microtissues. Finally, the 3D heart tissue was derived from induced pluripotent stem cell-derived cardiac muscle cells, which are able to beat like normal heart cells. All of these tissues were combined in an adapted microfluidic chip that was designed for the culturing of suspension cells in media, with several spatially separated solid tissue models which are shown in the middle. During the experiment, NK cells either stayed in the enrichment zones on the ends of the chip or circulated back and forth along the microfluidic channel via rocking, which also perfused the media, allowing for exchange of soluble factors between the three cell types. By using this method, the authors were able to assess a whole host of markers and phenotypes of how the NK, tumor, and cardiac cells behaved in the system. To predict which cells the NK cells would be able to recognize, the authors looked for ectopic expression of membrane-bound MIC-A/B. As you can see by these lovely images, the tumor cells had an abundance of this ligand, shown in red, while the cardiac cells did not, indicating that the NK cells should target the tumor cells for killing. So, to test this hypothesis in static, non-moving co-cultures, the authors measured the size of tumor and cardiac microtissues, and found that regardless of the growth cytokine treatments meant to supplement the NK cells, the tumors were continually getting targeted and destroyed. In fact, after 3 days, the authors confirmed that the tumor cells were completely degraded. Expectedly, the NK cells did not target the cardiac cells, and they were left morphologically intact. Now, this provides a proof of concept for MIC-A/B targeting, but because this experiment was done without cellular movement, the authors also needed to test this function in their model system. So they used multiple co-culture combinations where monocultures were used as a baseline reference, which is shown by the Venn diagram here. Of critical importance for this study is how the tumor cells grow. So when tumors were cultured by themselves or with cardiac cells, they found that over a 3-day period in 18 individual tumor microtissues, the tumor cells continued to grow unimpeded. But when NK cells were added to the mix, there was a heterogeneous change in tumor microtissue size. Overall, though, the growth of the tumors was much lower. For the tumors that shrunk in size, there was a higher NK cell accumulation in the tissue compartment and tumor itself, which correlated with higher cytotoxic activity and lower tumor cellular integrity. Curiously, the tumors that continued to grow in size had very few NK cells present. The authors attributed this heterogeneous tumor growth suppression to lower levels of interaction on the first day, and to the proteolytic shedding of the MIC-A ectodomain, which was found in the supernatant. Interestingly, the presence of any other human cell actually increased the shedding of this receptor by the 3rd day studied. Now, to fully understand what was occurring, the authors further visualized the integrity and infiltration of the tumors. In this representative image, they identified lots of tumor tissue integrity markers and few activated NK cells inside of them, indicating that NK cells are killing the tumors from the outside in. The authors next investigated the chemokine and cytokine signaling in different co-culture combinations in their system. They found that over the course of 3 days, the tumors in the presence of NK cells alone or in triple co-culture secreted increased pro-inflammatory IL-8, which is known to be a predictive marker of higher grade tumor burden and resistance to chemotherapies. Additionally, the authors measured multiple NK cell-derived cytokines, which act as markers of indirect anti-tumor activity. They found that NK cells secreted increased levels of GM-CSF, interferon gamma, and TNF-alpha. Interestingly, the interferon gamma and TNF-alpha levels spiked on the first day, but consistently decreased over time, in some cases down to basal levels. Finally, the authors investigated the behavior of cardiac microtissue during all of these cellular interactions. Using 4 exemplary cardiac microtissues, they found that the cells remained intact and did not express injury biomarkers, indicating that the NK cells and tumor cells were not structurally damaging the cardiac tissues. However, when looking at the cardiac cellular contraction profile, as it's important in normal heart beating, particularly in triple co-culture, the authors found increased time between the contractions, and a majority of the tissues exhibited irregular contraction amplitudes. These results correspond to known information which indicates that increased pro-inflammatory cytokines negatively affect cardiac contractility in vivo. Now, to quickly summarize everything all together, the authors used a microfluidic device to create an environment where NK cells and tumor and cardiac microtissues can be observed in culturing conditions. They tested multiple cellular configurations to identify specific effects of cells on one another. But the most interesting results occurred in the co-culture of all of the cells together. They found that this culturing setup decreased tumor growth while also causing increased pro-inflammatory secretion from the tumors. Furthermore, the NK cells secreted multiple pro-inflammatory anti-tumor cytokines, which is likely what caused the decreased tumor growth. All of this interaction did not have any effect on the structural viability of the cardiac cells, but did cause irregular contraction profiles, indicating that the immune tumor activity affects cardiac cells adversely. Now, not only do I think that these method developments are exciting to investigate and learn about, they're also significant in a broader context. This information is significant because it firstly illustrates that there are possibly adverse cellular reactions to adoptive cell therapies that we have not been able to detect. And by using a system that enables investigation of multiple parameters of cell function, a framework can be provided for more thorough investigations of how immunotherapies would function in patients. Ultimately, all science is basically a stepping stone for new knowledge, and these steps are driven by questions. And I had a few questions myself after reviewing this information. My first question revolves around the particular processes that are occurring through immune interaction. Because the authors were able to recover the cells after the experiment, are the tumor and NK cells able to be further analyzed to understand the cellular pathways by which the NK cells actively inhibit tumor growth? In regards to the system that they used, would it be possible to put multiple healthy tissues in the system as a readout, or even to possibly create some of these chips in series to investigate the multi-organ effect of localized tumor killing? Furthermore, to make this more applicable to people who are actually experiencing cancer, is it possible to make microtissues from primary cancerous cells and then put those in the system to see which therapies would work best for the individual? As always though, my final question revolves around you. What sort of ideas or questions popped into your head when hearing about this information? I would love to hear about them in the comments section below. Also, let me know if there are any topics that you'd like to hear about in the future. Ultimately, I hope that you learned something. But more importantly, I hope you enjoyed your time doing so. So if you did, give this video a like and subscribe for more in the future. Well, that's everything for today. Thank you for watching, and I'll see you next time.